The science is proven. The business model is not. By 2030, the global CAR-T market is projected to reach somewhere between $15 and $25 billion, or it may fail to scale at all. That range exists for a reason. CAR-T therapies save lives, but their complexity and cost mean only a fraction of eligible patients ever receive them. In 2025, there is broad agreement across biotech and pharma on one point. CAR-T works. In blood cancers, it has converted near-certain mortality into durable remissions, and in some cases, long-term disease control. But the real question is no longer whether CAR-T works. The question is whether it can scale from a boutique rescue therapy to something that treats thousands or tens of thousands of patients. And if it can, which model gets us there? Autologous, off-the-shelf allogeneic, or in vivo programming? In this video, we'll break these models down the way investors, payers, and pharmaceutical companies actually evaluate them. What scales, what breaks, and what ultimately gets bought. Welcome to BioLogicLab, where cell and gene therapies are analyzed as systems, not just science. The bottleneck isn't efficacy. Here's where CAR-T actually breaks. What makes autologous CAR-T effective is also what limits its growth. CAR-T consistently produces deep remissions in lymphoma and multiple myeloma, and early data in autoimmune diseases suggest similarly strong biological effects. When functional immune cells reach the right patient, the results can be dramatic, yet access remains limited. The reason is systemic. Three constraints dominate CAR-T deployment today. First, end-to-end cost. Not just list price, but manufacturing, quality control, logistics, hospitalization, and toxicity management. Second, time. Traditional autologous products require weeks from leukapheresis to infusion, and some patients simply do not have that time. Third, infrastructure. Specialized centers, trained teams, validated processes, and reliable cold chain. As a result, only a small fraction of eligible patients receive CAR-T. This is why the industry has converged on three competing models. Start with autologous, the current benchmark. Patient T cells are collected, genetically engineered ex vivo, expanded, and reinfused. It is highly personalized therapy. From a clinical perspective, its strengths are well established. Durable remissions are documented across multiple hematologic malignancies. Regulatory pathways are familiar. And because the cells originate from the patient, immune rejection is minimal. But the same features that enable efficacy also limit scale. Each batch is custom manufactured, throughput is low, costs remain high, cell quality varies with patient condition, introducing product inconsistency, and operations are fragile. A single manufacturing or logistics failure can delay or cancel treatment entirely. Autologous CAR-T defines what is biologically possible. It does not define what is operationally scalable. Then there's the industrial bet. Allogenic CAR-T replaces patient-specific manufacturing with donor-derived or iPSC-derived cells. These products are engineered once, produced in large batches, frozen, and stored as inventory. Operationally, this approach aligns more closely with traditional pharmaceutical manufacturing. Treatment can be delivered immediately. Batch production improves capacity utilization, and at scale, cost reductions on the order of 50 to 70% are theoretically achievable. However, the challenge shifts from operations to immunology. Allogenic cells face rejection. Persistence may be limited, and without precise gene editing, there is risk of graft versus host disease. Engineering complexity increases, often requiring multiple edits, which introduces new regulatory and manufacturing considerations. Still, pharma interest is real. Roche's multi-billion-dollar commitment to Poseida's Allogenic platform indicates confidence that these hurdles may be manageable. Allogenic CAR-T is fundamentally an industrialization strategy, with biology as the gating factor. The most disruptive idea comes last: in vivo programming. If this works, it changes everything. If it doesn't, it fails very visibly. Rather than manufacturing cells outside the body, genetic instructions are delivered directly into a patient's immune cells, often via a single infusion that turns the patient's own immune system into the factory. The potential advantages are significant. Manufacturing resembles that of a drug, not a cell product. Distribution becomes simpler. Costs could decline substantially, and outpatient administration may be feasible. But this model introduces a different risk profile. Targeting specificity remains an open challenge. Dose and expression control are more complex, and long-term safety must be demonstrated carefully. Regulators treat in vivo approaches as hybrids of gene therapy and cell therapy, extending development timelines. In vivo CAR-T is best viewed not as a replacement for existing models, but as a platform with high optionality and, correspondingly, high uncertainty. There is one variable that could completely redraw the CAR-T market. Early clinical data in lupus and neuromuscular autoimmune disorders suggest CAR-T may induce deep immune resets, in some cases allowing patients to discontinue chronic immunosuppression. If these results are confirmed, the addressable market expands dramatically from thousands of oncology patients to potentially millions of individuals with autoimmune disease. Critically, autoimmune indications may not require the same depth or durability of target cell elimination demanded in cancer, which could materially shift which CAR-T platforms are viable at scale. The implications for scalability, pricing, and platform selection are significant. We'll examine this in detail in a future episode. When you step back, a clear pattern starts to emerge. Autologous CAR-T delivers proven biology, but is operationally constrained. Allogenic CAR-T offers near-term scale if immunologic hurdles are managed. In vivo approaches promise the greatest long-term scalability, but with the highest control and safety risk. This explains current pharmaceutical strategy. Established autologous products generate near-term revenue. Allogenic programs are scaled where biology appears tractable. In vivo platforms are layered in as long-term options. This is not indecision, it is portfolio construction. The future of CAR-T will not be decided by preclinical curves. It will be decided by which models regulators approve, payers reimburse, and manufacturers can operate at scale. That is the focus of BioLogicLab, breaking down cell and gene therapy platforms into the models, incentives, and failure modes that matter to real decision-makers. Comment your pick and why below. Which platform do you think wins long-term? For deeper analysis of in vivo delivery, autoimmune expansion, and manufacturing economics, subscribe and turn on notifications. We'll cover whichever gets the most votes next. Thanks for watching.